Okay, welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effect seminar series. My name is Diego, and I'm a member of the VES Organizing Committee, along with Stephen Airwood, Mireya Selma, Jan Ilbudu, and Adrini D'Souza. These seminars are made possible with support and a lot of help from Laura Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas Variant Effects Alliance. So a huge thank you to them. As a reminder, we're always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I have some housekeeping items to mention. Each talk is about 20 to 25 minutes, so there should be time for some questions. Please post those questions at any time to the Zoom Q&A, and we'll read them out to the speaker at the end of the presentation. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can watch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. You don't have to worry about copying these URLs. I'm going to post all these links to the chat once we get started. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. And for the last housekeeping item, this is rarely an issue, but just as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So without further ado, we have two excellent speakers scheduled for today's presentation. First up, I'm very excited to introduce Florence Chardon, who is a PhD student in the Shendure and Starita labs in the Department of Genome Sciences at the University of Washington. The focus of her PhD is on developing novel multiplex single-cell and bulk functional genomics sequencing-based technologies to better understand gene regulatory architecture and the consequences of genetic variation in several different in vitro cell culture models. The title of her talk today is Multiplex Single-Cell CRISPR-A Screening for Cell Type-Specific Regulatory Elements. So Flo, go ahead and share your screen and start whenever you are ready.
Cool. So thank you so much, Diego, for the nice introduction. And hi, everyone. I'm super excited to tell you all about the work we've been doing in the lab. I primarily work with a postdoc, Troy McDermott, who's a postdoc in the Shendure lab. And we've worked together to develop this multiplex single cell method where we use CRISPR activation perturbations to identify cell type specific regulatory elements that can upregulate specific genes of interest. And so to start out, kind of why we're really interested in developing these kinds of methods is in the cases of haploinsufficiency, where a single functional gene copy is not enough to produce a wild type phenotype. So this can happen when you have a deleterious mutation in one of two gene copies, such that one of two alleles is no longer functional. And for many cases, this is actually not a problem and biological function can still occur as usual, but there are over 600 genes that are believed to cause disease when haploinsufficient. So it's a really interesting area to study, and specifically, we really want to take advantage of the fact that there is still one remaining functional allele that we can utilize for CRISPR perturbations. And so the idea here is that we can use a CRISPR activation perturbation at the remaining functional copy to basically use that copy to increase transcription off of that allele. And if you do this successfully, you can actually rescue the phenotype and bring gene expression levels back to normal. And so we know that this works, but to be able to do this really successfully, you really need to know exactly where to target, whether that's a promoter element or an enhancer element. And before I go into some of the work that I'll talk to you about today, I wanted to go over two really beautiful examples that kind of show why we're so interested in doing this on a really large scale. So the first demonstration of using CRISPR activation to rescue these phenotypes was back in 2019. This work came out of Nadav Ahituv's lab at UCSF, and they were studying this gene SIM1, which leads to an obesity phenotype when you have a deleterious mutation in one of the two gene copies. And so they were able to show that when they target CRISPR activation to actually both the promoter and enhancer of SIM1, so separately in different experiments, they were able to rescue this phenotype from both of those perturbations. So this was the first demonstration of using CRISPR activation to rescue a phenotype like this. And this obviously was amazing that they were able to show that it works in a mouse model. And then more recently, this just came out last year and it's currently a bioRxiv paper. This is primarily out of Kevin Bender's group at UCSF. They showed that when they targeted the SCN2A promoter, they were also able to rescue electrophysiology deficits in mice. And so these two examples are amazing that they were able to show that using CRISPR-A, they can bring these mice back to wild type biological function. But for both of these pieces of work, they're studying one gene and kind of optimizing guide RNAs for specific regulatory elements regulating these genes. And so we thought, we could take what we've learned from this kind of work and develop a really highly scaled method so we could identify guides for many different genes potentially at the same time. And so that brings me into the introduction of the project that I'll talk to you about today, which is this really high throughput method by which we can screen many different guide RNAs at once. We can target many different regulatory elements, so both promoters and enhancers. And the whole idea is that we can screen many elements at once and identify candidate guide RNAs that can potentially rescue haploinsufficient disorders. And then I also want to mention that kind of an unintended benefit of this kind of screening framework is that we're also able to identify enhancer gene pairs. And the reason this is really exciting is because there's so many enhancers regulating so many different genes, and there's so much that's not known in this area. And it's not because of a lack of effort, it's just really hard to understand, which sequences are regulating which genes in different cell types and in different times in biological development. And so here I'm just going over briefly kind of the main pieces of this work. So the first kind of key aspect of this work is that we're multiplexing CRISPR-A perturbations in single cells. And so I'll talk about two screens we did today. One was in K562 cells, the other was in iPSC-derived neurons. And in both of these screens, we're trying to get as many guide RNAs as possible into a given cell. And the reason for that is because the single cell RNA sequencing readout that we use is still quite cost prohibitive. So the more guides we can get per given cell, that just increases the power in our experiments without an associated increase in cost. And when we put these guide RNAs into cells, the cells are already expressing the CRISPR-A machinery. So that's the dCas9 with a variety of transcriptional activators, and we used a couple in our different screens. And then after we do the wet lab portion of the experiment, we do the data analysis in which we're looking at cells that have a given guide RNA versus cells that don't. And then we just look for an associated increase in expression, both for the target gene that we're intending to target, and also all genes within a 1 megabase window upstream and downstream of the target site. And then we ask, are we getting specific upregulation of a specific gene within this defined window? And for our CRISPR-A perturbations to be really useful, we want them to be specific. So we actually don't want them to be upregulating more than one gene, because if we want this to be therapeutically relevant, you of course only want to upregulate the gene that you're intending to upregulate. And so on this slide, I'm going over the guide RNA library that we used in the experiments that I'll tell you about today. So you can see that the library is heavily biased towards targeting these 9 prioritized neurodevelopmental disorder risk genes. And the reason we have hundreds of guides targeting these 9 genes is because we wanted to target all annotated and known promoters of these genes. And so another thing to mention is this is also a large collaborative effort with a couple of other groups who are then able to take some of these candidate guide RNAs and test them in vivo mouse models. So these are the genes that we're really interested in finding successful guides for. And then we also had 100 guide RNAs that act as non-targeting controls in this screen. So those guides should not have an effect at all. And then there's 2 other classes of guides that we tested. One is just transcription start site positive controls. So we would expect for those to see an increase in transcription. And then kind of a neat piece of this library was testing 50 guide RNAs that target 25% previously annotated K562 enhancers. And I'll go into the data a little bit on that later, but we were really interested to see if we could target a CRISPR-A perturbation to a distal enhancer and see upregulation of the intended target gene. And so before I go into the data, I just want to briefly discuss the vector that we used for these experiments. So traditionally, CRISPR screens actually use lentiviral vectors, and those are actually like really great and nice to work with because a lot of cell types are very easy to transduce, and you can very tightly control the number of guides that you're getting in an individual cell. That would have worked for our K562 experiment, but we were really interested in porting this over to a more relevant cell type for the genes that we're specifically studying. And so we chose to move this into an iPSC-derived neuron framework. And with many differentiating systems, silencing is a huge issue of viral vectors. So we chose to use a piggybac transposase-based vector to kind of ameliorate the silencing issue and be able to get stable expression of guide RNAs in our neuron-derived system. And there's kind of two key pieces of this vector that I want to highlight. The first is that we're getting a functional Pol3 transcribed guide RNA off of the U6 promoter. So this is the piece of the vector that's actually doing the CRISPR perturbations. And then we also have the second transcript, which is a puromycin resistance cassette fused to a GFP. And this GFP allows us to sort for GFP expressing cells. And I'll talk a little bit later about why that's helpful for our experiments. And so, as I mentioned previously, we did this screen both in K562 cells and iPSC-derived neurons. For the K562 screen, we tested two different activators, the VP64 activator and the VPR activation complex. And then in the iPSC-derived neurons screen, we used the DCAS9 VPH activation complex, primarily because we had this iPSC line with that CRISPR activation complex integrated at a safe harbor locus, which again is really important because of the silencing issue that you get during differentiation protocols. And so here's a more kind of detailed outline of what the screen looks like. So after we've cloned this guide RNA library into this piggybac vector, we then transfect this library along with the piggybac transposase into the two different K562 cell lines that we used. And then we do 10x single cell transcriptome profiling after having allowed the cells to grow out for a period of 9 days. And then after this, we do our differential gene expression testing, where we just look for each guide RNA. We look 1 megabase upstream and downstream of this target site. And then we simply ask, did this guide RNA lead to an expression in any genes within this window compared to cells that didn't have that guide RNA? And if we see a significant difference, then we can conclude that that guide RNA is having an effect and is able to upregulate that gene. And so I'll show the results for all of these screens in the form of a QQ plot. So on the x-axis is the expected p-value distribution. So this is the distribution you would expect if there's no effect at all. And then on the y-axis, I'm showing the observed p-values in our experiment. And so these are the results from the K562 screen. And we were really excited to see that we're seeing significant upregulation from many of our targeting guide RNAs. So the presence of these extremely significant p-values tells us that there's signal in our screen. And clearly some of these guide RNAs are working really well because we're not seeing the same effect when we're testing non-targeting guide RNAs. And then I mentioned previously that we wanted to test whether we could target enhancers to see upregulation of their target genes. And one reason we really wanted to do this was because the literature on targeting enhancers with CRISPR-A is a bit conflicting. Like, it's definitely possible, but then there also was recently a paper suggesting that it might be really important to co-target the associated promoter. But we saw in our screen that when we target these enhancers alone with a CRISPR-A perturbation, we actually are seeing upregulation of quite a number of these target genes. So that was an exciting result from this screen. And then to wrap up the K562 portion of the talk, this just shows the hits in the screen and how many of them were specific and upregulated genes that we predicted that they would. So most of them were. There were 3 non-specific cases of upregulation in K562 cells. Two of these, I think, really are non-specific. The COM3 one is an interesting case because the guide RNA targeting the intended gene for that targets this promoter that actually looks like it's bidirectional and like it could regulate COM3. So that one, we're not clear if it's really an off target or it might just be a promoter that's also regulating COM3. And then on the enhancer side, we got pretty specific upregulation as well, except for two cases in which targeting this enhancer actually upregulated ASIC1, which is a different gene than was previously coupled with this enhancer. And then the HMGA1 enhancer is actually a potential novel enhancer gene pair finding because this was previously not an annotated enhancer gene pair. And so after we did the K562 screen, which was really kind of a proof of concept of, does this assay work and can we see upregulation of genes in this screening framework, we wanted to move this into a more relevant cell model for these specific neurodevelopmental disorder genes that we're really interested in identifying functional guide RNAs for. And so we moved this whole framework into an iPSC-derived neuron framework. And so I mentioned previously that in this cell line, the CRISPR-A complex is integrated at a safe harbor locus to avoid silencing during differentiation. And this is a pretty neat system where upon the addition of a single small molecule, the degron that normally degrades this complex is stabilized, and then you get expression of the CRISPR-A complex. And then here is just a nice image showing that the CRISPR-A system works in these mature neurons. I won't go into the details, but essentially we're just testing whether a single guide RNA can activate a TD tomato reporter that's driven off of a minimal promoter in this system. So this is just to show that the CRISPR-A perturbation is working in neurons. And after we did that validation experiment, we went ahead with our actual CRISPR-A screen. So the framework and workflow is pretty much identical to the K562 screen, except the only difference is now we're transfecting into iPSCs. And then, of course, we have to do the differentiation into neurons, which we did for a period of 19 days. And then after that, we do the same 10x single cell transcriptome profiling. And I mentioned previously that we can actually sort on GFP to get cells with higher numbers of guide RNAs. And this is just to show you what the data looks like when we don't sort on GFP. The median and mean is just showing the number of guide RNAs per cell. And here on the right histogram, this is the data from sorting on the top 40% of GFP. And you can see that our median number of guide RNAs per cell goes from 2 up to 5. So sorting on these GFP positive expressing cells, like the highest fraction, proportionally boosts the number of guide RNAs per cell, which is really nice because it allows us to increase the power in these experiments. And then the analysis strategy is the same. We again test for differential expression of genes within a megabase upstream and downstream of the target site. And then we ask, which guide RNAs are leading to appreciable upregulation of their target genes. And then one thing we wanted to do before going too far into the data analysis is ask whether the neurons that we're generating are really mature neurons as we think they are. So the data here is from a different paper from Barbara Treutline's lab, where they did this really nice single cell transcriptome time course looking at this exact differentiation protocol over the course of 35 days. And what you can see is that there's distinct transcriptional states that are arising during this 35-day time course. And so what we were able to do was co-embed our single cell transcriptome data with this data set and basically see how it compares. So here I've highlighted our cells in blue, and we see that they really nicely overlay with the mature neuron cluster from that time course. And then you'll probably notice that some of our cells do seem to be stuck in an intermediate state, and that's likely due, we don't know because we didn't systematically test it, but the major difference between our two protocols is that they add glial cells to their culture, whereas we don't add that, and we believe that that might be the main difference here. And so I'll show the results in the same way as I did for the previous screen in this QQ plot. And again, we see that we observe the presence of these highly significant p-values when we look at our targeting guide RNAs versus the non-targeting control guide RNAs. And then here, On the right is the same exact plot. I've just zoomed in the y-axis from 0 to 50 to be able to see the resolution a little bit better. We see that for most of our targeting guide RNAs, we are observing upregulation of the target gene. And then I just wanted to briefly go into a couple examples that are really cool from this screen. So this is probably my favorite example. So TBR1 is a gene that is normally expressed like in neurons and in most organisms, but in this specific NGN2 induced neuron system, TBR1 is actually known to not be expressed. And then in K562 cells, when we targeted the TBR1 promoter, we don't see upregulation at all. So when you look at the p-value from this perturbation, it's the minus log 10 of the p-value is close to 0, showing that we're not really seeing significant upregulation. And when you look at the chromatin state of the TBR1 promoter, it looks to be largely closed. And then when we make the same perturbation in the iPSC-derived neurons, TBR1 was a hit in this screen, and we see that we're getting really significant upregulation of the TBR1 gene. However, when we look at the chromatin context of the TBR1 promoter, it still looks to be largely closed, but this is a really neat example where we can potentially target this region of closed chromatin and see upregulation of TBR1. Which is really neat. And there's so much that's not known about CRISPR-A mechanisms specifically. So kind of anything that we learn from these screens is super useful for the CRISPR screening field, and also to better understand how these regulatory regions are regulating these genes. And then I just wanted to show what the enhancer data looked like in the iPSC-derived neuron screen. So I showed you in the K562 cells, we're able to target these enhancers and see upregulation of these target genes. And since we use the same guide RNA library in the neuron screen, we can directly compare the results. And when we target these enhancers in the neurons, we don't see a single case of target gene upregulation. And these enhancers are K562 specific enhancers, so we wouldn't expect to see upregulation in the neuron screen, but the fact that we don't see any at all, I think just speaks to the power of doing these screens in the relevant cell types, because specifically for studying enhancer elements, we know that they are cell type and tissue specific, and kind of seeing these contrasting results between these two screens is a really neat example of studying these enhancers in the right cell types. And then this is the last kind of locus example I wanted to show you all, which is basically looking at how the chromatin landscape might explain the cell type specificity that we see of the enhancer-mediated upregulation. So here I'm showing the ANXA1 enhancer that looks to be largely an open chromatin, and we're able to see upregulation in K562 cells. And then when we look at the chromatin profiles in the iPSC-derived neurons, there's not any major peaks in this region, and we also don't see successful upregulation in the neuron screen. And so to finish off the talk, I just kind of wanted to summarize what the results looked like in both the screens across the different classes of guide RNAs that we tested. So when we look at the positive controls in this screen, which were those guide RNAs targeting transcription start sites, we see a pretty good correlation between the K562 and the neuron screen. Then when we look at all of the promoters that we targeted, that correlation is still there, but it's a little weaker. And part of that, if you remember, is because we targeted all known and annotated promoters of these genes, many of which are not going to be used in the two cell types that we tested. So some of these guide RNAs aren't having an effect because they're not targeting a promoter that's in use in these cell types. And then the most striking result from this screen was the total lack of correlation that we saw when targeting enhancers. And that's because we're targeting specifically K562 enhancers. So we see that those work in the K562 screen, but we see that they don't work at all in the neuron screen. And so to summarize, our CRISPR screens, they capture a signal from known enhancers in K562 cells and from these risk gene promoters, both in K562 cells and iPSC-derived neurons. This framework really allows us to identify candidate guide RNAs that can be followed up on and potentially used for rescuing haploinsufficient disorders that are caused by haploinsufficiency in these genes. And then we really envision that this framework can be deployed to discover guide RNAs that are capable of treating the over 600 diseases that are associated with haploinsufficiency and low gene dosage. And so that's the end of my talk. I mainly want to thank Jay Shendure, my PI, that Troy and I have both worked with a lot on this project, and then also a couple of people in the Shendure lab that have helped out a lot with experimental techniques and discussions about the data analysis, Beth, Sam, Sylvia, JB, and Diego. And then this project is a larger collaboration with Nadav and Stefan Sanders and Nick Page, and then Leah Starita, who's my other co-advisor, and then a big thanks to Martin Kampman, who gave us the iPSC line that we used for the neuron screen. And with that, I'm happy to take questions.
Thank you very much, Florence, for this great presentation. There is some applause. The first question from the audience comes from Ten Yuang. How do you assign perturbation effects of a particular gene in a multiple single guide RNA per cell design?
Yeah, so that's a really good question. So we do multiplex multiple guides per cell, but the way that we do the data analysis is we go one guide at a time, and we basically look at all of the cells in our screen, and we put them into these two groups. So cells that have that guide and then cells that don't. Then the cells that have that guide, those cells, of course, also have other guides that are unrelated to the guide that we're testing. But because we're introducing these in random combinations, it's actually okay that there's other guides in that cell targeting other genes, because the way the screen is designed is we're basically underpowered to detect any kind of combinatorial effects. And that basically allows us to just test one guide at a time and then look, for this group of cells that all have this one guide, what kind of upregulation are we seeing in the vicinity?
Okay, thank you. The next question by Matthew Coelho. Really beautiful data. I was wondering how easy it was to tease apart the effect of individual guide RNA bioinformatically. When you have 5 guide per cell with GFP sorting and the Piggybac system. Out of 500 guide RNAs, many of them are predicted to have effects.
Yeah, so that's kind of related to the answer to the previous question. So yeah, we're underpowered to detect combinatorial effects. The other thing is for these screens, so we aim to have a couple hundred of cells harboring each guide RNA to be able to detect certain effects. But of course, if you're having, like, say you have other guides in the cell that are having an effect at some low level, and that specific combination is present a couple times, we're just not seeing it enough to detect that. So we really are able to look at individual guides and determine, if they're having an effect or not. And then, yeah, so that's kind of the answer for, oh, sorry, it seems similar to the last question. Yeah.
All right, I think we can move to the next speaker.